Assalamu alaikum wa rahmatullahi wa barakatuh. Bismillahir Rahmanir Rahim. Welcome to today's biology class. That is the third session for the chapter 11, Reproduction in Organisms. So in the previous second class, we have learned about what is called inflorescence, and then based on the type of inflorescence, there are two types, cymose and racemose. And then we have also learned we can categorize flowers on the basis of their mode of pollination. Some are insect pollinated, wind pollinated, water pollinated, and animal pollinated flowers, and we have discussed the different characteristics of them. And today inshallah we will discuss about two phenomenon. They're called microsporogenesis and megasporogenesis. That means the production of male gametophyte and the production of female gametophyte inside the flower. So how these gametes are produced, that is called microsporogenesis and megasporogenesis. And inshallah we will also discuss about fertilization process in plants to some extent. So at first we describe about the process of forming male gametophyte, that is male gamete. And this process is called microsporogenesis because we say that male gametes are called microspore because they are the spores which are small in size. That is why this process of formation is called microsporogenesis. And we know that genesis means the way that something is produced. And also we say megasporogenesis, that means the process of forming megaspore, that means female gamete production process. As the female gamete is larger in size, that is called megaspore. So when we say about how the male gametes are produced inside the flower, at first we observe this figure, and from the book I have merged two separate figures into one so that we can understand this process clearly. So we start from the anther. We know that inside a flower there is androecium, and inside the androecium there may be multiple number of stamens, and inside the stamen there are anthers. So inside the anther there is a colony of cell called microsporangium that contains numerous pollen mother cell that is diploid 2n. So when we take an anther and make a cross section on it, we will find some colonies of cells. These colonies, each of them are called microsporangium. So here we see that from the cross section of anther we get 4 microsporangium, and actually microsporangium are the sac-like structures which contains the microspores. And inside the microspores, inside the microsporangium we find numerous number of pollen mother cells. So we have zoomed to only a single pollen mother cell, and we know that this pollen mother cells undergoes meiosis cell division, and it produces 4 haploid daughter pollen cell or microspore. So this pollen mother cell diploid undergoes meiosis to produce 4 daughter pollen cells or microspores. So each pollen cell, when they get matured, they looks like this. Their outer surface is somewhat spiky and sticky also. And when the pollen cell is pollinated by some way, either by wind or by insect, when this pollen cell falls on the stigma of a carpel, then the pollen cell undergoes some change. The nucleus of the pollen cell divides mitotically to produce 2 nucleus. One nucleus is called tube nucleus and another is called generative nucleus. So here we see that when the pollen falls on the stigma of a plant, so what happens then? So here you can assume that the red marked part is the stigma of a flower. So when pollen falls on it, the nucleus of the pollen cell divides mitotically to produce two nucleus. So the big nucleus is called tube nucleus and the small nucleus is called generative nucleus. And also here we see a pollen cell has two membranes. The outer membrane is called exine and the internal membrane called intine. So the intine, the inner membrane, elongates to produce pollen tube. What happens? The intine or inner membrane that is increased in size and that moves downward, so it produces a path through the stigma and the style, and this path is created by the tube nucleus. The tube nucleus pushes the membrane of the intine to make a path, and this path is followed by the generative nucleus. So that pollen, so inside the pollen tube, the tube nucleus remains at the bottom. It makes the way, and this way is followed by the generative nucleus. And during this travel, in this meantime, the generative nucleus divides into two male gametes. So while that is traveling through the tube, in the meantime, the generative nucleus divides mitotically to produce two male gametes. So that is how we see how a male gametes are produced in the flower and inside the pollen. So we have finished the process of microsporogenesis or male gametophyte. So here we see some of the process is completed within the anther, and some of the later changes takes place when after pollination. So now we will discuss about megasporogenesis. That means the production of megaspore. That means the process of producing female gametophyte. So we observe this figure. So I have also merged two separated figures together to make this, and this figure shows that how megaspore are produced inside the ovule, and then how the ovule undergoes some change to produce ovum and secondary nucleus. So we start from the ovule of the flower. So inside the ovule, megasporangium, the sac contains single megaspore mother cell that is surrounded by nucellus tissue, and by meiosis cell division it produces 4 haploid megaspore. So at first we start from the carpel, and inside the carpel there is ovule, and here we see that the ovule, this green part is called the nucellus tissue that surrounds the megasporangium. So inside the megasporangium we find only a single megaspore mother cell that is diploid, and it will be divided by meiosis process to produce 4 megaspore. So here we see that inside the ovule 4 megaspores haploid have been created. And now from the 4 megaspores, 3 disintegrates, that is damaged, and only 1 megaspore at the bottom survives, that remains, and then it develops into embryo sac. Then it undergoes some changes. So we have seen that inside the ovule we have got 4 megaspores by meiosis, but among these 4 megaspores 3 will disintegrate, 3 will get damaged, and only 1 megaspore will survive. And this single megaspore will turn into embryo sac, and inside the embryo sac some change will take place. So what are these changes? The megaspore cell's nucleus divides to ultimately produce 8 nuclei by mitosis, and residing 4 at each pole. What happens is that the nucleus inside this embryo sac, there is only one single nucleus. So this single nucleus divides mitotically to produce at first 2 nucleus, then 4 nucleus, and then 8 nucleus. So here we see that at the 2 opposite poles of the ovule of the embryo sac, there are 4 and 4 nucleus located at the opposite poles. So then among this 4 and 4, 8 nuclei, 2 nuclei will come at the center and will produce a structure called secondary nucleus. And so here we see that at the opposite poles now there are 3 and 3 nuclei present. So this 3 and 3, 6 nuclei will turn into separate cells. So they produce some cells. Here we say that this pole is actually near the micropyle, near the pore of the ovule. So near the pore of the ovule or micropyle, the middle cell, the cell at the middle, it turned into egg or ovum, and the two cells surrounding the egg cell are called synergid cells. And we have seen that these two nuclei fuse together to form secondary nuclei and secondary nucleus. And at the opposite pole there are 3 cells present. These 3 cells are called antipodal cells. So what we see now, inside the embryo sac there are 3 antipodal cells, and at the center there is a secondary nucleus, and near the micropyle the cell at the middle is called egg or ovum, and it is helped or assisted by 2 synergid cells. So we can understand the process of megasporogenesis and microsporogenesis by observing a video now. So let's see the video. This video describes about the pollination process, the fertilization process, and also the megasporogenesis and microsporogenesis before it in a flowering plant. So we will first describe about the megasporogenesis that takes place inside the ovule. So this white marked boundary part is called ovule. So we have said that earlier the ovule contains a single megaspore mother cell that is undergoing meiosis to produce 4 megaspores. But among them 4 megaspore, 3 will get damaged and only 1 will survive. So this only 1 megaspore will form embryo sac, and the nucleus of this embryo sac and the nucleus of this megaspore divides mitotically to produce at first 2 nucleus, and then 4 nucleus, and then 8 nucleus. So among these 8 nuclei, 2 nuclei will come at the center to form secondary nucleus, and the rest 3 and 3 will go to the opposite poles of the embryo sac. So now this nuclei turn into complete cell. So these 3 cells are called antipodal cells, and the cell that is red marked here near the micropyle is called the egg or ovum, and that is assisted by 2 synergid cells. So by this process megasporogenesis has been completed. Now we will talk about microsporogenesis. So inside anther we have said many functions, many steps of the microsporogenesis have already taken place. So after this process, we have found numerous mature pollen cells. So we will first start from a mature pollen cell here. So the pollen cell contains a nucleus. This nucleus undergoes mitosis to produce a tube nucleus and a generative nucleus, as we see in this video. So this mature pollen will travel by air or by any insect, and that will fall on the stigma of a carpel. So when it falls on the stigma, we have said that the intine, the internal membrane of the pollen cell will increase in size and will penetrate through this stigma and style and will move downward, and this path will be followed by tube nucleus and then by the generative nucleus. So this tube makes a path and travels downward to reach to the ovule through the pore of micropyle. So during this travel, the generative nucleus turns into two male gametes. So one male gamete will fertilize the egg or ovum, and another male gamete will fertilize the secondary nucleus. So here we see that the fertilization process has been done two times. The fertilization takes place for the secondary nucleus and also for the egg or ovum. That is why this process is called double fertilization. That means two events of fertilization takes place simultaneously, and the double fertilization process is a unique feature of angiosperm plants. Usually gymnosperm plant don't undergo double fertilization. So by this process we have understood how microsporogenesis, how megasporogenesis and fertilization process takes place inside a flowering plant. So now you have a homework that describe the process of microsporogenesis and megasporogenesis with figure. The figure I have provided in this video. I suggest that to draw this figure to learn. No more today. সবাই ভালো থেকো ইনশাআল্লাহ। আসসালামু আলাইকুম।